Today we will discuss about difference between healthy pancreas and the pancreatic cancer, or the pancreas that have some kind of cancers because of the mutations in the pancreatic cells. So we will start with the healthy pancreas. So these are the actual images of the healthy pancreas by staining the pancreas, and these are the pancreatic cancer cells or pancreas that has the pancreatic cancers. Those are stained with this uh, staining agents, and this is for an overview that these are considered as the pancreatic cancer cells, and these are the healthy pancreatic cells. So this information shows about the healthy pancreatic cells, and this figure shows about the pancreatic cancer cells, and we can see that the cells are not rounded in shape in the case of PDAC, and also the populations of the cells are very low compared to the case of this healthy pancreas. So this indicates that pancreatic cancers might be more migratory in nature. That's why they are not able to found too many pancreatic cancer cells at a small uh, at a particular time. So one should not confuse that because the amount of cells is low in PDAC, pancreatic cancer cells replicates or does not have this uncontrolled growth rate characteristics. These are more aggressive cells compared to the other cancer types. So let us just zoom in into this healthy pancreas and this pancreatic cancers, and this is the schematic view of what is actually going on in the healthy pancreas. We can see that the laminin are there. Laminin is a glycoprotein that helps in the uh, towards triggering various cell functions, and we can see that amount of laminins are sufficient in the case of healthy pancreas. Looks like this uh, sufficient in the case of healthy pancreas, whereas the amount of laminin in the case of the pancreatic cancer cells are very low. We can see that they are very low. So in the case of healthy pancreas, the interactions is occurs between cell to cells, and whereas in the case of pancreatic cancer cells, the interactions mainly occurs from instead of occurring from cell to cells, they occurs with the help of the integrin that are present on the cell surface. And because of that, the contractile cytoskeleton in the pancreatic cancer cells can interact with the ECM, and this connects the ECM with the cytoskeletons of the pancreatic cancer cells, and in the that helps in the migration of the pancreatic cancer cells from one place to another place, or from one part of the body or one part of the organs to the another part. And apart from this, these are the pancreatic cancer cells, which are and we can see that the stromal layers, that is the region surrounding the pancreatic cancer cells, are also increased, whereas in the case of healthy pancreatic cells, everything is uniform, and everything is uniform, and this laminal plays a role towards interacting with the cells. So these are the cells here. The four cells are shown. So laminal plays a role towards interacting with the cells, and the adhesion junctions helps in the interactions of the two cells. Whereas since the pancreatic cancer cells here directly interact with the ECM, so the ECM helps in the coordination of this movement of the pancreatic cancer cells. Whereas in the case of healthy pancreas, it's the laminal adhesion junctions more predominantly plays the roles towards the cell movement. And one more thing, that pancreatic cancer cells, we can see the fibroblast formations. This is uh, because of the because of the proteins that are secreted by the pancreatic cancer cells that leads to the change in the tumor microenvironment, and those tumor microenvironment also leads to the formation of this fibroblast. Other things are also changed, such as hyaluronic acid is increased in the case of pancreatic cancer cells that act as a that helps in the immune suppressions and also act as a pro-tumorigenic factors for the pancreatic cancer cells. And the collagen formations. The collagen formations, and because of the tumor microenvironment, it act as a pro-tumorigenic factors, and one of the things that promote these pro-tumorigenic factors is the high collagen fiber formations. So in the case of pancreatic cancer cells, we can see the high collagen fiber formations, and the crosslinks are also increased. So that will act as a physical barrier that will prevent the immune cells to come and come and find out the non-self epitope on the pancreatic cancer cells. Whereas in the case of this healthy pancreas, all these collagens are very uniform, and it does not disturb the regular functions of the pancreatic cells, healthy pancreatic cells. These are just a basic overview and difference about the healthy pancreas and pancreatic cancers cells, or the pancreatic cancers environment. If you need to understand more about these differences in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.